Horizontal cells are the laterally interconnecting neurons having cell bodies in the inner nuclear layer of the retina of vertebrate eyes. They help integrate and regulate the input from multiple photoreceptor cells. Among their functions, horizontal cells are responsible for allowing eyes to adjust to see well under both bright and dim light conditions. Horizontal cells provide inhibitory feedback to rod and cone photoreceptors. Retinal neurons include photoreceptor cells, horizontal cells, bipolar cells, amacrine cells, and ganglion cells. Structure. Depending on the species, there are typically one or two classes of horizontal cells, with a third type sometimes proposed. Horizontal cells span across photoreceptors and sommate inputs before synapsing onto photoreceptor cells. Horizontal cells may also synapse onto bipolar cells, but this remains uncertain. There is a greater density of horizontal cells towards the central region of the retina. In the cat, it is observed that A-type horizontal cells have a density of 225 cells/mm2 near the center of the retina and a density of 120 cells/mm2 in more peripheral retina. Horizontal cells and other retinal interneuron cells are less likely to be near neighbors of the same subtype than would occur by chance, resulting in exclusion zones that separate them. Mosaic arrangements provide a mechanism to distribute each cell type evenly across the retina, ensuring that all parts of the visual field have access to a full set of processing elements. MEGF10 and MEGF11 transmembrane proteins have critical roles in the formation of the mosaics by horizontal cells and starburst amacrine cells in mice. Function. Horizontal cells are depolarized by the release of glutamate from photoreceptors, which happens in the absence of light. Depolarization of a horizontal cell causes it to hyperpolarize nearby photoreceptors. Conversely, in the light a photoreceptor releases less glutamate, which hyperpolarizes the horizontal cell, leading to depolarization of nearby photoreceptors. Thus, horizontal cells provide negative feedback to photoreceptors. The moderately wide lateral spread and coupling of horizontal cells by gap junctions measures the average level of illumination falling upon a region of the retinal surface, which horizontal cells then subtract a proportionate value from the output of photoreceptors to hold the signal input to the inner retinal circuitry within its operating range. Horizontal cells are also one of two groups of inhibitory interneurons that contribute to the surround of retinal ganglion cells. Illumination \display style \to center photoreceptor hyperpolarization \display style to horizontal cell hyperpolarization \display style to surround photoreceptor depolarization. The exact mechanism by which depolarization of horizontal cells hyperpolarizes photoreceptors is uncertain. Although horizontal cells contain GABA, the main mechanisms by which horizontal cells inhibit cones probably do not involve the release of GABA by horizontal cells onto cones. Two mechanisms that are not mutually exclusive likely contribute to horizontal cell inhibition of glutamate release by cones. Both postulated mechanisms depend on the protected environment provided by the invaginating synapses that horizontal cells make onto cones. The first postulated mechanism is a very fast ephaptic mechanism that has no synaptic delay, making it one of the fastest inhibitory synapses known. The second postulated mechanism is relatively slow with a time constant of about 200 milliseconds and depends on ADP release via pannexin 1 channels located on horizontal cell dendrites invaginating the cone synaptic terminal. The ecto-ADPase NTPase1 hydrolyzes extracellular ADP to AMP, phosphate groups, and protons. The phosphate groups and protons form a pH buffer with a pKa of 7.2, which keeps the pH in the synaptic cleft relatively acidic. This inhibits the cone Ca2+ channels and consequently reduces the glutamate release by the cones. The center-surround antagonism of bipolar cells is thought to be inherited from cones. However, when recordings are made from parts of the cone that are distant from the cone terminals that synapse onto bipolar cells, center-surround antagonism seems to be less reliable in cones than in bipolar cells. As the invaginating synapses from horizontal cells are made onto cone terminals, the center-surround antagonism of cones is thought to be more reliably present in cone terminals.
Please subscribe and thanks for watching.